Good morning, good afternoon and good evening. It is with great pleasure that I welcome you all here today for this webinar on novel genetically encoded contrast agents for preclinical optoacoustic imaging. I'm Dr. Tim Devling, Director of Sales and Applications at iThera Medical, and I will be moderating this session. The webinar itself will last for about 40 minutes and we really encourage questions. If you maximize the Go-To tool, you can see a question box where you can type them in and we will answer them at the end. The webinar will also be recorded and we will send you a link in the next few days. Our speaker today is Dr. André Stiel, leader of the Cell Engineering Group at the Institute for Biological and Medical Imaging at the Helmholtz Zentrum, Munich. André has a long history with protein engineering for imaging. He gained his PhD in the field of photoswitchable proteins for super-resolution imaging with Stefan Hell at the Max Planck Institute for Biophysical Chemistry in Göttingen. André continued his work in imaging and report development before starting the Cell Engineering Group at the IBMI. Here the focus is on the development of unique reporters for high sensitivity optoacoustic imaging, which is what he will speak to you about now. So over to you, André.
Yes, welcome everybody, and thank you, Tim, for this kind introduction. Actually, Tim smuggled in the word novel here in the title. What I rather try to do today is to give you a more or less comprehensive overview about what happened in genetically encoded contrast agent for optoacoustic, because I believe that is, and I will tell you later in more detail, a very promising avenue for whole animal imaging. All right, as Tim already said, my background is in fluorescence imaging and specifically super resolution imaging. And there I developed photoswitchable proteins. These are GFP-like proteins, and I will tell you a bit later about them. And this made me basically grew up in a world of beauty, so to say. So as most of you, I think, are aware of, that fluorescence imaging here, just a random assortment of images, has the possibility to visualize a number of targets with different colors and in high resolution, giving us all those beautiful images. And one factor I believe that made the success of fluorescence imaging as a life science method was the availability of genetically encoded reporters, meaning biological reporters that you can basically only by putting the gene in your cell of interest have the reporter molecule. And that allows you to be cell specific, to have basically no dilution and no perturbation of your imaging experiment, in contrast to, for example, dyes that you have to add from the outside. And when we look at the publications of fluorescence microscopy or fluorescence imaging here in gray, overlaying with publications on the green fluorescent protein, which was basically a subclone for use in imaging in 1994, we see that the strong advancement of this imaging technique coincides more or less with the introduction of this genetically encoded labels, despite the imaging being around much longer. So I believe the genetically encoded fluorescent proteins really have a key to fluorescence imaging. But now, how is it if we go to in vivo imaging, like, for example, animal imaging, which is still a method for us to understand most about an organism and also interactions in the organism? So we obviously have intravital multiphoton microscopy, and this allows us to use all these tools that are existing for fluorescence microscopy, and also allows us to do multiplexed imaging here, different cell types and structures in the tissue. But intravital microscopy has a very small imaging volume compared to other imaging methods. And that allows us to only see a small glimpse of, for example, the whole pathophysiology in a mouse. Now, on the other side, we have fluorescence whole animal imaging. And there you can see, obviously, the complete mouse. But due to light diffusion, you have only a very poor resolution here in the mimicking here. This allows you not to detail any interactions of cells or distributions in special regions. And obviously, it's additionally surface weighted so that you only see basically an accumulation of the signal throughout the animal. And the same is true for bioluminescence imaging. Now, here optoacoustic comes into play, because with optoacoustic, we can basically enhance our imaging volume massively while still retaining a quite scalable higher resolution due to the readout of ultrasound. And we could image, for example, a complete mouse and here with endogenous contrast see the vessel structure of the mouse. So for those of you who are not quite familiar with this method, let me briefly explain you. So you have a laser excitation of a given chromophore, and in this excitation, like in any excitation process, you absorb a photon to an excited state of the molecule, and what then happens is basically decay of the energy to the ground state via vibrational relaxation. And this vibrational relaxation results in nothing else than basically a microheating of the environment of the molecule, and by this a micro expansion, and this expansion is nothing else than a sound wave, and this sound wave can be detected as ultrasound by ultrasound transducers, giving us a readout for optoacoustic imaging. And that is already a fundamental point of this method, that the photophysics underlying it are essentially similar to fluorescence imaging. In fluorescence imaging, we read out the decay in fluorescence. In here, we read out the decay in vibrational relaxation. Now, what can be those contrast agents? So the method is most famous to date for reading out endogenous contrast, like for example, blood contrast coming from the heme molecule in the blood. And there, I'm sure a lot of you know this really successful imaging, for example, of vessel structures in dermatology, but also of blood oxygenation and blood flow in general, indicating, for example, inflammations or indicating metabolic disorders. And when we come now from endogenous contrast to targeted contrast, we obviously have synthetic targeted contrast, like nanoparticles or organic dyes that have to be added to the animal from the outside and targeted with different techniques. And we have the topic of today, we have transgene contrast. And I like to separate transgene contrast basically in two categories. The one is a transgene contrast encoding enzymes that basically catalyze the production of a reporter, of a label, of a chromophore, of a pigment, you could say. And this you can then use in optoacoustic as a contrast agent. And the second class is where the gene itself directly produces a protein, and this protein is immediately the reporter. So the chromophore is there within the protein. You will see later more about that. All right, and this directly brings me to one of the challenges and complexities of targeted contrast in optoacoustic, and especially of transgene contrast, because the endogenous contrast from blood is on the one side really a virtue, because we can basically see all the vessel structures and also can see other biomolecules like lipids, for example. But on the other hand, that poses basically a very strong background for all other targeted contrast agents that we want to see. And while with nanoparticles and with dyes, you can find chemical ways to create really a lot of contrast and also mask these agents in certain areas, for transgenes where you are limited by the capabilities of biology, by the capabilities of the cell, so to say, it is always a challenge. And due to this, researchers try to focus on something called the blood window here, roughly beyond 650, 700 nanometers, where the signal from blood contrast is relatively low. But, and this makes me, so to say, a believer, the fact that with those transgenes, we have to address the photophysics that we also know from fluorescence imaging, but also the general photophysics of chromophores. And you know from nature that there are plenty of chromophores, flowers, fungi, bacteria. You will see later about that. And this is in strong contrast, at least in my opinion, to basically all other whole animal imaging methods that might be capable of this topic. I mean, MRI, PET, bioluminescence, and ultrasound, they all have certain ways of transgene reporters, or semi-transgene in the case of PET, and some are really ingenious, like the bubble formation in ultrasound. But still, they are relatively complex, and they use contrast mechanisms that are not natural in nature, I want to say. And the use of photophysics in contrast is really something that nature uses also for light reception, for coloring, and so on. And so I believe that the use of optoacoustic imaging using photophysics is really the key to us moving in the life science direction. And due to this, I believe that the now so successful imaging of the vessel structure, which is routinely done in optoacoustic imaging, will be basically enhanced in the next decades, hopefully, with the imaging of cell distributions and also of the function of cells with a resolution that truly can inform us about biological and pathophysiological processes. With this said, I want to start to tell you a little bit about those transgenes. Let's start with the first class, where an enzyme cascade basically encodes the pigment. I think the successful, or the most successful to date, is the melanin. Melanin is produced by the expression of tyrosinase. Tyrosinase is a rate-limiting enzyme that mostly converts from tyrosine, the more or less structured polymer melanin. And this polymer is basically ubiquitous. It is in bacteria, it is here in fungi, but also in the human skin, as most of you know. And what researchers quite early, starting from 2011, did was to encode the human tyrosinase, for example, in tumor cells, and then use this contrast from the produced melanin to basically identify those tumor cells. And here a very nice image from the Bird Lab, where they see a tumor, and down to this small clusters are around 1,000 cells. So in context with the vessel infrastructure. So this signal is very strong for melanin, but the challenge, from my opinion, is that the spectral signature of melanin is just a slope, and that makes unmixing of the signal, especially in a noisy environment, quite complicated. And another factor, that at least in my experience, and so far I haven't seen too much to the difference, is that the overproduction of melanin is not something that makes the cells very happy. And due to this, we see most work with cancer cells, or with cells that are anyway supposed to be short-lived, or with inducible systems like here with doxycycline. So what other pigments could be used? For example, there are a number of bacteria that produce pigments for light harvesting, or for shielding, or for coloring. One is the Chromobacterium violacinum, and the Westmeyer lab could encode the enzymatic cascade that produces this violacin pigment in E. coli bacteria, and then inject those bacteria here in tumors, and could also resolve this in optoacoustic imaging. But you can also already see that the peak of the violacin absorbance is quite far out of the blood window. My lab used bacteriochlorophyll also as a reporter. So bacteriochlorophyll has advantage that it's far out in the blood window here, very nice, and with a double peak signature. But again, here, the catalysis mechanism to form those bacteriochlorophyll chromophores and to embed them in the protein structures is very complex. And there comes a challenge. It's hard to translate those to mammalian systems. And that is what we eventually want, because we want to label every cell type of interest in our animals. A shortcut was done here by the Wang lab by using the Laxet gene. And Laxet converts basically a dye called XGAL to an indigo dye, or a cage dye to basically the indigo dye. And they could show here the signal in the brain. And it is rather nice, and it also is in the blood window. But the problem is that you always have to inject those substrate for the dye into the imaging area. And this is directly the reason why pigment productions are maybe not the road to where we want to go. Because either you have to basically provide the substrate, or you really complex enzyme cascades to produce those. And there are plenty more colors in nature, from plants, fungi, and the bacteria again. But often these pigment pathways are extremely challenging. They are often secondary metabolites and need multiple enzymes. And due to this, the direct translation in mammalian cells on a high level, that you really can produce enough agent to discriminate it, might be very challenging. That brings me to the second class, where the transgene directly bears a chromophore. The most famous proteins in this class are definitely GFP-like proteins that we know from fluorescence imaging. And there we have a protein structure called a beta barrel with a chromophore in the center. And the remarkable thing about this protein is that it forms the chromophore. I don't go into detail here, but these are 3 standard amino acids from basically the protein. And due to an inherent catalytic machinery, those amino acids form the green chromophore and can also form other colorful chromophores depending on the amino acid surrounding. So now GFP-like proteins in optoacoustic, obviously they have been tried very early, already in 2009 by Daniel, then in Alicia Christos' lab. So they try to use directly M-cherry, a protein that is very successful in fluorescence imaging. But obviously the absorbance of this protein is really far out of the blood window. Might be a reason why here they could only show work in zebrafish. And ongoing works then compared different GFP-like proteins here, beautiful work from Laufer, where he compared different fluorescent proteins in their optoacoustic signal. But what you can always see, it's basically a cutoff at around 600. And this could also not being changed by really engineering proteins for optoacoustic imaging, engineering fluorescent proteins for optoacoustic imaging, here by a collaboration from the ZEMP lab with the Campbell group, where they search for a protein that has optimal optoacoustic signal by basically having no fluorescence quantum yield. But they could also not shift out the absorbance of the protein out of the strong blood absorbance. And what we see here are, again, bacteria pelleted in the ear, so the contrast is not very strong. So that makes me believe that with, for example, one of the reddest fluorescent protein, TEC RFP 657, was having an absorption of only 611 nanometer, that the absorbance of those proteins is inherently limited by the possibilities of the catalytic machinery. If you remember this beta barrel, what I showed you before, there are only a number of degrees of freedom for the catalytic machinery. And it's already very remarkable that the color variants really cover from almost UV to the near infrared. But I believe there is a limit at some point. And this is why with GFP-like proteins, we are stuck slightly in the blue from the blood window. Luckily, there is another protein class. This protein class, most prominent here, the bacteriophytochromes, are porphyrin chromophore-bearing proteins. So porphyrins are this 4-ring structures here, and they have absorbances far in the near infrared. And the good thing is, despite the fact that they cannot be autocatalytically formed by this protein, so they must be attached, one of the most used porphyrins here, biliverdin, is a catabolism product of heme. And that means it's present in most mammalian cells. And if not, it can be also enhanced in number by co-expressing some of this mammalian enzymes here to produce a biliverdin. So bacteriophytochromes have been popularized by the Vakusha lab starting in 2012. And here we see IRFP 713. And this already has a strong absorbance right perfectly in the blood window. And they could see here tumor cells labeled with IRFP rather deep in a mouse in vivo. A second class of porphyrin-bearing proteins that was explored by my lab for optoacoustic is fucobilly proteins. So inherently, and that was why we looked into those, fucobilly proteins have the advantage that they can take up to 3 chromophores in one protein structure and can even have more. I will tell you later about that. And the challenge is that the first biliverdin binding fucobilly protein was absorbing quite far out of the blood window at roughly 600 nanometer. And what we did, we really engineered the chromophore surrounding to push it more into the blood window here with roughly 690 nanometers. And there we could see bacterial cells labeled with those proteins subcutaneously. And why we only show bacterial cells? Because the chromophore binding of those proteins is still rather challenging. So the attachment of this biliverdin chromophore so far works much more reliable in bacteriophytochromes, maybe because they have already been much longer around than in fucobilly proteins. So if you would ask me now what would be the best conventional NIR reporter for optoacoustics, I would normally refer to IRFP 720 from the Vakusha group. It has a quite nice absorbance in the blood window, and so far in most of our comparisons, it was always the best near-infrared conventional reporter. But with this near-infrared reporters, we solved the issue that we are more in the perfect region of the blood window, but still we have to overcome the contrast from the blood, right? So, and when we not want to look at tumors, and before in almost all examples I showed you tumor cells, but when we want to look at really biological cells of interest, like immune cells that are only present in low amounts, then mostly the signal from our reporter in the imaging experiment is far below the signal from our tissue background. And that makes it really challenging to image this with conventional reporters. So in 2015, we presented a way around that. And this is taken from fluorescence imaging, where this concept is already known for quite a while. So what you do is basically you change the state of your label from an on state, having ultrasound emission, to an off state, and you can change between those two states by light of two different wavelengths. And what you then do is you illuminate your animal with off switching wavelengths, here pulse, 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 pulse. With this, a reporter signal goes off. And then with the second wavelength, you bring it on again and then switch it off again. And with this, you can create a modulation of your reporter signal. And obviously, your blood background and all the other background does not have a modulation. And due to this, you can simply separate your reporter signal from all background, making basically the background invisible. And beyond that, due to the fact that the kinetic with which these transitions happen are characteristic for different molecules, you can also multiplex by distinguishing different switching kinetics. So how does this work, and how can we genetically encode it? There are transgene proteins actually in both classes, bacteriophytochromes and also GFP-like proteins, that show this reversible switchable behavior called photoswitching proteins. And on the molecular level, this is nothing else than a cis-trans isomerization of connecting bridges here between the C and the D ring, and here between the 5 and the 6 ring of the GFP chromophore. And on the photophysical level, what that means is that we have here the normal process here for let's say the cis state. We have an absorbance, and then we would have some non-radiative decay, vibrational decay, to give us our optoacoustic signal. But we can also have another decay pathway, and this induces a cis-trans isomerization, basically a chemical change of the chromophore. And that brings us to a second state. And this second state, the chromophore, obviously has a different surrounding, because it's in a slightly different position, and a different amino acid surrounding directly results in different photophysical characteristics. And that again means that you can excite here with a different wavelength, that, for example, also the non-radiative decay is a little bit less, so that would then be the off state. But due to this excitation with a different wavelengths, you can switch back, also again a cis-trans isomerization, to your original state. And with this, you can switch back and forth to make the picture complete. Due to the fact that these are obviously molecular states, you can you also have a natural relaxation between those states. So just very briefly, why I'm also so very fond of those molecules, because they are with me now since over 15 years, because they're also used in super resolution imaging. There you use this off-on switching to basically quench all the fluorescence in your diffraction limited fluorescence emission with this famous donut-shaped beam that is the off-switching light. And what you left over with is a not diffraction limited spot, and that then gives you a super resolution image. Let's come back to optoacoustics. So what can we do in an optoacoustic experiment with this? So the simplest approach is differential imaging. You take an image when the protein is fully in the off state, and then you switch it off, and then you take an image fully in the in the on state here, and then you take an image fully in the off state, and you repeat this process. And with this, the Wang group in 2015 could already show 3,000 cells in 8mm depth labeled with this reporter protein. So that's already a remarkable low number. So the Laufer group made popular a slightly different version of this differential imaging by having an intermediate step So you have an enhanced signal-to-noise ratio, and here also they could nicely visualize the expression of this reporter here again in tumor cells with a Fabry-Perot imaging approach. With this, I want to come basically to the approach that we follow and started in 2015, where we basically not disregard all, like here, basically we disregard everything that happens in between. But obviously you can also record an image for each of the pulses of your off switching. And by this, you can take the complete photo switching kinetics, again, repeat this process, and then use different methods to delineate this. And the strong advantage here is that you can also use this to discriminate between different types of labels more easily, as I showed you at the beginning. And we showed that in 2015 in a quite simple experiment where we distinguished blood from one variant and from another variant of photo switching protein, depending on the photo switching kinetics. And in 2018, the Vekusha lab, together with a Wong group, could show the remarkable low number of 500 mammalian cells in a voxel with this technique. And in 2000, we could show that you can use this multiplexing with the different kinetics here, for example, to distinguish 3 different populations of reporters in a tumor environment, 2 Eukaryote cell populations and 1 E. coli population. And we could even see the mixture of different reporters. And here is a zone of mixture, mixing those and mixing those. And if we look at the molecular details, how does this work? So this here are structures of the chromophore of, let's say, slow reporters that, for example, would comprise one of those. And what happens here is that you have a rather strong interaction between this aspartate and the D-ring of the chromophore. And then in the faster variant of this reporters here, there is an arginine basically interacting with this aspartate here, and with this reducing the interaction to the biliverdin D-ring. And we believe due to this D-ring is less stabilized, and as you remember from earlier, the D-ring is basically what moves, and this is trans-isomerization. And we believe that due to this variant is a faster variant. And this directly tells you that with amino acid surrounding of those proteins, you really can play a lot and really tailor the photo switching kinetics, and with this populate, so to say, the colors in this case, which are the kinetics, to really allow multiplexed imaging of genetically encoded reporters and optoacoustics. So coming back to our work, what maybe is additionally important for the people really wanting to use those proteins is that we do not use high-end optoacoustic setup, but really commercial off-the-shelf M-sort from Arthera. And with this could also have this distinguish here between 3 different label populations, or here follow the growth of tumor sites from day 3 to day 14, really starting off with also around 10,000 cells following each individual site. But the sensitivity even yields down to here Eukaryote cells, and this is also another thing. Before we only talked about cancer cells; these are then lymphocytes, and there we're coming in the direction of really also biological interesting targets and also biological interesting numbers. Not completely there yet, but moving in this direction. And that we can get there shows this phantom experiment here. In this case, in the lowest concentration we have in a blood phantom, we have 63 cells per microliter. That is less than a cell per imaging voxel. Obviously, this is a very defined situation in a phantom, but this makes me believe that with this photo switching technique, we can really go in the direction of below hundreds of cells for optoacoustic imaging in vivo and also multiplex, and really following biological phenomena. So how does it work really in the lab? So we have an illumination schedule, it's called. That means, let's say, 10 times the off-switching wavelengths, 10 pulses, 760, then 10 times the on-switching wavelengths, and then we repeat this process 20 times. What we get then out is a time series of images, and when we analyze then individual pixels in those images, we can see basically the signal trace of these individual pixels. Here I only showed the pulses with off-switching light, And then we can do different simple operations with this. We can, for example, take the mean, then we see much nicer this off-switching, and could then, for example, use to distinguish different types of labels, or we can go in the frequency domain and see the repetitive behavior here. But obviously, it does not always look that beautiful. This is how, let's say, a normal trace, more deeper in the mouse looks. But the important thing is that we can still go by the mean and by the Fourier transform and identify this as a photo-switching reporter. And the important point that, in my opinion, distinguishes from spectral unmixing is that this photo-switching schedule is completely set apart from the rest of the experiment. It's a preset, and it does not interfere with your other noise parameters or your background. So with this becoming more and more important, let me, for the last minutes, look a little bit at two other, from my perspective, quite important developments that we do in the lab. So the photophysics of the labels, as I could show you now several times, becomes obviously more important the more you want to tune it, for example, to have different kinetic parameters for multiplexing, or also want to increase the contrast levels. And due to this, to understand the contributions of the different decay pathways, what we developed is what we call a multimodal optoacoustic spectrometer, where we read out the absorbance, the fluorescence, and the optoacoustic signal of a sample at the same time, and or shortly after each other. And I don't want to go in detail here, but what might be interesting for some of you is the very simple approach that we undertook to have quite good and accurate readouts by, next to the sample chip, placing a reference chip here, for example, filled with a dye that absorbs in the region of interest of your sample. And due to the time of flight inherent to optoacoustics, we have two different signals stemming from the same laser pulse that we then can use to correct the sample image, the sample signal, and with this have a very accurate reading. And the second topic that I briefly want to touch is that we need more defined test targets. So of all the examples from the different groups, and also my group that I showed you, we always compare different things. Obviously, we try to compare things that are accurate and also try to compare things that look rather remarkable. But what we really also would need is a way to compare things to say, okay, this is really that much better than the other one. And in this direction, what we try to do is use alginate beads. So this concept is already existing for quite a while. So what you do there, and we made a very simple contraption here. It's a syringe in a 50 ml falcon, and this here is filled by alginate, and this is by calcium chloride. And then drops of this alginate, when you centrifuge it, fall in the calcium chloride solution and solidifies. And you can mix this alginate with cells of interest that express your reporter molecule of interest. And what you then get are alginate beads filled with your cells. And the nice thing is that they are very defined. So they have a very defined size and also very defined fluorescence signal. And also by using different needle diameters, you can have different sizes of this alginate beads. And we could, for example, use those, inject it in here a tumor to create really a perfect, in this case, I think 200 micrometer point source that you then can, for example, use to compare different reporter types, because you have, again, this very defined situation of number of cells in a given volume. But also, for example, it could be interesting to further improve reconstruction and unmixing algorithms, because obviously these reporter or these molecules here have a very defined shape, and what ideally should come out is also a similar defined unmixed and reconstructed image. With this, I want to finish and have a brief look at what the future might, from my perspective, entail. And obviously, I'm a strong believer, as I think you could see in photo switching, and I believe that we will also see some advances in the way how we can analyze the data, and also in the type of reporters with stronger signal, different switching characteristics, and so on. And another concept that might be interesting is enhancing photophysics, as I like to call it. So obviously in transgene labels, we're always limited by the amount of protein we can express in a cell and when it becomes toxic to the cell. And due to this, it is interesting if there are possibilities to basically enhance the signal by photophysical interaction between the chromophores. And due to this, you need protein structures that allow you to have those interactions in a very defined way. And here, those phycobilly proteins might become interesting that I showed you earlier, because they come from bacteria where they form antenna complexes with hundreds of chromophores. And such protein macromolecules could result in a signal increase, and that, for example, eventually combined with photoswitching could also give us again an edge. And another topic that definitely also will become interesting are sensors, which are very popular in fluorescence imaging. So basically your signal is depending on the binding of a molecule, and Daniel's group and also the Wang lab could already show the use of fluorescence sensors in optoacoustic, and also the latest developments of the near-infrared sensors from the Campbell and the Kusha group, I think steps in this direction, that eventually we can also see the function and the molecular surrounding of cells in optoacoustics. With this, I want to finish with a picture from the lab, which is actually chemical quite inaccurate, but psychological correct. So I'm always really happy when I see something shiny in the lab. There are some postdocs which did most of the work that I showed, and obviously I'm indebted to Vasilis, Jill, and also former IBMI members Louis and Daniel, who definitely helped to kickstart the photoswitching in optoacoustics for me and for our institute. And with this, I thank for your attention, and I'm open for questions. Thank you.
Well, an excellent talk, André. Thank you very much. We have had a lot of questions coming in, so I will not wait any further and I'll start asking. So I hope you've got your thinking hat on. How do these bacterial chromophores compare to the newer fluorescent proteins? You've described the differences between IRFP and so
Sorry,
on.
Tim, I don't catch you properly. Can you speak louder, nearer to the microphone, something?
Sorry, can you hear me now, André?
Yeah, again, try it.
I'll try again. Is that better?
Yeah, that's better.
So, how do these bacterial chromophores compare to the newer fluorescence proteins? You've described some comparisons with IRFP, but what's your general feel in terms of sensitivity and stability and bleaching?
In terms of photoswitching versus normal bacteriophytochromes, is this what you mean?
Or fluorophores, yes.
Yeah. So the classes of proteins are similar. The bacteriophytochromes, actually the native bacteriophytochromes are already photoswitchable because they are photosensors in plants. And for the use in fluorescence, they started to make non-photoswitchable variants. In general, you could say that molecules that are non-photoswitchable tend to bleach more. So our experience is that the faster the photoswitching, actually the better the resistance to photofatigue. And the reason is that they can basically leave the excited state faster. And in our latest paper, we have some comparisons on the photofatigue. And in comparison to, let's say, normal bacteriophytochromes, these photoswitchable proteins are better. And this is also necessary because obviously we need this number of photoswitching cycles to achieve this discriminatory effect. So from my perspective, I think the talk made that clear. I strongly believe in the increase of sensitivity with photoswitching because it's just moving from the spectral domain to the temporal domain, where in theory, you can have unlimited sensitivity by switching just endless times, so to say. And you could now argue that with having spectral unmixing, you could also average multiple times. But there also your noise floor is still always dependent on the same parameter. But this photo switching pattern really is something extrinsic. And due to this, I believe that it always will be a stronger discriminator than spectral unmixing.
Okay. So in respect to the actual photoswitching proteins, One question here is, could you comment on the difference between the different phytochromes you're developing? And I suppose you've kind of just answered this, but what future path do you see towards developing better phytochromes for photoacoustic imaging?
Mm-hmm. So at the moment, I think that the complete field of this photo-switching phytochrome development is very in its infancy. We have, I think at the moment, 5 or 6 phytochromes from the different groups that are almost in all cases engineered on the level of truncations, meaning you make them smaller and you try to find those truncation, as also we did, that it does not or even make it better in a photophysical sense. But so far, the direct surrounding of the chromophores in terms of amino acids, I showed you this image earlier of the 2 structures, has been almost not touched. And there I believe that we will definitely be able to engineer a number of different kinetics variants that makes this multiplexing easier. And you could argue that if you have fast imaging systems, also with faster switching, you could accumulate more discriminative power in a shorter time. So that might be also interesting. And obviously, by playing with the direct surrounding of the chromophores, there might also be a stronger absorption cross-section, although there I believe that we are almost in the area where it ends. I think at the moment it's around 120,000. And there is one bacteriophytochrome reported by actually my former group in Göttingen for stat imaging, where they reach an absorption cross-section of almost 200,000. And so more I never saw, and maybe we will reach also with the photo switching proteins that level. And obviously you always try to have more absorbance, but I think again there is a chromophore photophysics that at some point put a limitation to it. And there, that's why I showed you the fucobilly proteins. There it might become interesting to have multiple chromophores in one protein and then have other enhancing effects, so to say.
Cool. I think some people have jumped on the kind of difference between the your ability to distinguish different chromophores and the kind of temporal characteristics you're looking at here. So one question that has come up a few times now is, what is the temporal resolution when using your photo switchers? So are you losing out really in terms of temporal resolution?
Mm-hmm. Absolutely, perfect question. Because yes, you do. I mean, because you need the repetitions for discrimination, and the more noisy your environment is, often the more repetitions you need to be discriminative. So you always need this time to get basically your discriminative effect. And with this, you clearly lose out in the time resolution as opposed to single shot imaging. This is clear, and I would say at the moment with the MSOT, we are in the range of minutes as time resolution. But obviously, I mean, if you have a faster laser repetition rate, so what I showed you in the images is not resolved in time, but resolved in laser pulses. So it only depends on how quickly you can deposit the energy that basically induces the switching. And if you use faster switching systems or faster laser systems, you can also switch faster. And due to this, I believe that time resolutions below the seconds are possible with obviously optimized systems, but it will never be a real competitor to one-shot imaging. On the other hand, the question is, how good can you image other reporters with one shot?
Okay, that's a very good answer there, Andre. One follow-up question from this is, what typical energy are you depositing per pulse? Is it essentially a special laser? Well, I mean, the answer is no, but I'll let you answer that.
No, I mean, as you already said, Tim, so that is, I think, the one of the key take-home messages here. You can obtain, for example, our reporters, but I'm sure also other people from Transgene. And when I don't make advertisement now, but obviously when you have an MSOT at home, you can directly use this, at least our type of imaging, with this machine. And there we have energies around 70 millijoules. And obviously, yes, I mean, the lower the energy, the more it becomes like this. And obviously, the nicer it becomes like this, also depending on the label expression, obviously. But there is no special trick beyond the normal imaging, at least to the imaging I showed from us. But when you look, for example, at the paper from the Wang group from 2015 and at the paper from 2018, where they use more or less the same system, but with technical improvements, you can very nicely see that better laser repetition rate and so on definitely can do a trick, yes.
Cool. A couple of questions on the biology here. So did you notice any immunogenicity or biocompatibility problems with the proteins that you expressed? Or was there any negative reactions within the animal? I'd probably say.
No, so far not. On the caution side, obviously, I have to add here that most of our work so far was, let's call it, in the context of imaging scientist experiments, meaning not really following very intricate biological phenomena where certain conditions or behavior can be observed, like, for example, that some immune cells are still perfectly functioning like they did before. But I'm a firm believer that we will have at least less trouble here than, for example, with the expression of complete enzyme cascades for pigments, because fundamentally those bacteriophytochromes are not too different, and in quotation marks, from GFP-like proteins. And there we have experience since 20 years, and we know that there are challenges. Nobody can say that expression of a GFP protein does not perturb a cell, but it perturbs us comparably little to other approaches of engineering contrast. And this, I believe, is the same with bacteriophytochromes and also with the photoswitchable labels bacteriophytochromes that we are using. So we use them in Eukaryote cells already, and they are quite successfully, and also in some other immune cells, where I speak on another day. And there the cells behaved normally. So we didn't have early cell death or something else. No.
Okay, and probably a question that most people will be thinking about, and that is, can you comment on the correlation here between linearity of cell number and signal detected?
It's rather hard to go directly from signal detected, because we are basically having the discrimination from the unmixing process. But I wouldn't say it's completely linear, but as I could show here, we and then the problem, and not the problem, let's say, the point here is that we use basically more a classification than a signal-based unmixing. So we classify all those as reporter present, and we could also look at the signal itself, and that more or less is linear in the bounds of experimental systems like this. But the discrimination effect also depends on a number of other points, like, for example, movement correction, what I didn't talk about now, also the noise level, and so on. And all this together will give you the discriminatory effect. And if this then really follows a strictly linear form, this I cannot say now, but what I can say is that we can reach relatively low limits, and that intrinsically it can go to single cell level. I put this out now with a lot of caution, but from fundamental point of view. And if this can be reached on a linear level, it's something else.
Brilliant. Thank you for taking the time for all of the questions. We do have some more, but what we'll do is we'll answer them offline individually. There are quite a few here. And I just want to say thank you very much, André, for giving a wonderful talk. Some people have been asking, these reporters or the plasmids are available from Adgene if you want to use them in your own lab, or contact André directly. I'm sure he'll appreciate any collaborations. And I'll just say again, thank you for the talk, and thank you all to the audience for taking the time to spend here with us today.
Thank you, Tim, and bye-bye, everybody.